Yeah. Other participants, please put your camera on because every day we have to take the photograph along with the speakers. We have to take the photographs.
जी जी
Akshay, then Samir, Avinash, Dr. Avinash.
Yes, sir.
So, Dubal, good morning.
Good morning, sir. Good morning to all.
Good
morning,
sir.
to
How
all.
are you? Morning, sir. How about cold there?
Very, very good morning, sir.
Good morning. Good morning.
Okay, Namaskar, and once again, very warm good morning to you all. As I said, today, two eminent speakers are going to speak on their interesting topic, and they will put their thoughts. Today is the 8th day of our training program. Today, first speaker, Dr. Junjar B. Dubal. He is working as principal scientist in the Division of Veterinary Public Health, ICR, IVRI, Ijatnagar, Bareilly, Uttar Pradesh, and sir is going to talk on effluent treatment plant for disposal of solid and liquid slaughterhouse waste. And today another eminent speaker, respected Dr. S.B. Barbude sir. He is working as director ICR at National Research Center on Meat, Hyderabad. And sir is going to talk on different herder technologies used for improvement of meat quality for export purpose. So now around 115 participants have joined, and we will start the today's session. I request our professor, Dr. Jhende sir. He is the PI and course director of this training program. I request Jhende sir to please give brief introduction about our today's first speaker. So Jhende sir, please.
Yeah. Thank you, Dr. Vaidya. Very good morning to everyone. Very warm welcome to all. Today is the 8th day of the our training program. I have pleasure to introduce today's first guest, Dr. Jhadvi Dubal, senior scientist working at ICR. Principal scientist is working at ICR IVRI in the division of the veterinary public health. I am proud to say that he is our student. He graduated and post-graduated from our department, veterinary public health. And after that, he did his PhD from IVRI. I joined at IVRI as a scientist, and is working since 12 to 15 years, having working experience. And after PhD, Dr. Vaidya has, Dr. Dubal has excelled in the different techniques in the division of the VPHI and working hard in various areas of the research in the zoonosis, then meat hygiene, milk hygiene, environmental hygiene, antimicrobial resistance. These are the is specialized area in which he is working on different organisms for the AMR profiling. So far, Dr. Dubal has guided a 5 MSc student for their postgraduate studies and one PhD student uh, for the PhD degree program in the IVRI. Initially, he worked at northeast part, that is the NEH region, for almost 9 years. And after that, he also joined at Goa ICR Center and worked for the 2 years. And after that, he is now working at IVRI. So throughout his research career, he has established a very high level of the commitment and involvement in a few national projects, as well as the invited participations in the expert groups in AMR, that is Antimicrobial Resistance, and is actively involved in organizing various symposiums of IAVP, that is Indian Association of Veterinary Public Health Specialists, and also the international conference, that is ISOPAL 2013, which was held in Goa. So far, he has completed 4 research projects as a principal investigator and also involved as a co-PI in various projects at IVRI, and also helping the students which are coming from other states for studying over there. He has received many awards as an best research paper presentation award, best oral presentation, and also the best poster presentation awards. Till that, he has published more than 40 research papers in highly reputed national and international journals. In addition to his popular articles, book chapters, extension folders, and other accession numbers for publishing various genes. He has also evaluated and reviewed the many international and national research papers and master and PhD degree thesis. In addition to, he is also working as an associate editor for the Journal of Veterinary Public Health, which is released every year every quarter by our Indian Association of Veterinary Public Health Specialists. So, with this short brief introduction of the Dr. Dubal, I invite Dr. Dubal to deliver his today's talk on effluent treatment plant for the disposal of the solid and liquid slaughterhouse waste. So, Dr. Dubal, please.
Thank you very much, sir. Good morning to all of you. And first of all, I would like to thank the organizer, Dr. R.J. Jhende, sir, as well as Dr. Vilas Vaidya. I would like to thank our Honorable Vice Chancellor, Dr. A.M. Paturkar, also for providing me the opportunity to speak on the today's topic. So I am sharing my presentation. So is this visible?
Yes.
Yeah. Thank you very much. So today's topic that is on the effluent treatment plant for the disposal of solid and liquid waste which are generated in the slaughterhouses. This is very interesting topic and very important pertaining to the environmental safety point of view and for the utilization of the waste to make the proper slaughterhouse byproduct. Though I am new in this topic, but I collected a lot of information, and for me it was very interesting, and I try to compile in a better way. So we will start with the slaughterhouses. We all know the slaughterhouses is the places where the animals are killed. But actually the meaning of abattoir is to have the modern facilities where the human slaughter can be carried out, and it also includes the hygienic processing and preservation of wholesome meat, and the ultimate aim of this abattoir is to produce the safe and wholesome meat. But the another basic aim of the abattoir is also to hygienically dispose and treat the solids and liquid waste which are generated in the slaughterhouses. But if we see the developing countries as well as the our India, we have a lot of slaughterhouses, and these are not up to the mark, and these are obsolete as well as unsuitable and unhygienically carried out the most of the operations. So meat produced from these slaughterhouses, they may release the diseases or they are the produce the contaminated meat. That's why a report released by the Central Pollution Control Board, they have pointed out the need of the upgradation of the old slaughterhouses to the modern one, so that with the objective of the human slaughter and production of the safe and wholesome meat, processing of this meat in hygienically way, as well as the effective separation of clean and dirty slaughtering operations. It also ensures the safety measures and utilization of edible as well as inedible byproducts. And another objective that is the important for the today's topic, that is the environmental safety through the treatment and disposal of the waste and effluent. So these slaughterhouses waste are nothing but the effluent. Why it is not? Just a minute. So these slaughterhouses, these entirely depending on the slaughtering capacity, which is measured as the livestock unit. So one livestock unit is the one cattle or two pigs or two three cows or the five sheep. So generally in the small slaughterhouses, up to 30,000 livestock units has been killed per year, and this is generally carried out in the small villages, panchayates, communities. Medium slaughterhouses have the capacity of the 50,000 animals, while the large abattoir have the more than 1 lakh livestock unit. The majority of the slaughterhouses in India, they are illegal, but beside this there are 1,176 slaughterhouses, and we have the 75 modern slaughterhouses, and we are the leading in the meat exporter, as the second world largest exporter, but we have only 75 modern slaughterhouses. So we produce large amount of the slaughterhouses waste, which generally contain the organic molecules or the organic materials, suspended solids, and the waters, which consist of the colloidal forms of the fats, proteins, and cellulose. So up to 24% of the water which are used in the food and beverage industry, that is mainly coming from the food industries. So these slaughterhouses is nothing but the effluent which has the potent pollutant sources, which contains the high organic water, and it contains the solids and semi-solid material like the blood, stomach contents, manures. Besides this, some pathogenic organisms as well as non-pathogenic organisms are also present in these slaughterhouses waste. Besides this, the water which is used for the cleaning and washing purposes, and therefore large amount of this disposal, this need to be treated very effectively to avoid the environmental contamination, to spread of the to reduce the spread of the contagious or zoonotic diseases, and the ultimate aim is to preserve our natural resources like rivers, ponds, and soils, etc., and for the economical benefit to utilize the byproduct. So these effluents are coming from the larrages, slaughtering operations, bleeding, then the dressing and processing operations, and these are categorized as the solid, liquid, and gaseous waste. Solid waste includes the bone, grease, feathers, manures, grit, then the indigested ingesta, hairs, aborted fetuses sometimes. Liquid consists of blood, urine, water, dissolved solids, stomach contents, and gas, that is the odor and emission. So about 45% of the soluble and 55% of the coarse suspended organic matters are present in these slaughterhouses waste. When these are released into the water bodies, they can create many problems, like it inhibits the light penetration to the aquatic animals. It also encourages the eutrophication, and ultimately it leads to the deoxygenation of the rivers, so that the oxygen content or the dissolved oxygen content will be reduced, which ultimately affects the aquatic life as well as the plant. And sometimes the anaerobic stage of oxidation can be takes place, which further increase the nitrate and ammonia in the water body, and ultimately it also leads to increase the algal bloom. So these are the harmful And beside this, some arsenic, these are generally used in the poultry industries to increase the weight or prevent the parasites, as well as to give the skin color. And these also some of the responsible source for the cancer and the skin diseases. Besides this, as we know, the antibiotics which are commonly used, particularly in the poultry industry, some poultry industries, not in all, and these also causes the antimicrobial resistance problem. Actually, the topic, this antimicrobial, as I am working on the antimicrobial resistance, it itself take a lot of time to discuss on the antimicrobial resistance. So this is not the today's topic. So, but beside this, some diseases also spread in some tropical countries like the cholera, typhoid, malaria, dengue, fever, dysentery. Some pathogens like Campylobacter, which has generally originated in the poultry carcasses, then the Salmonella, Yersinia, E. coli, STC, then the hepatitis E viruses. These are also produced from the slaughterhouses. So beside this, the recent outbreak, if we know the SARS, then the SARS-CoV and COVID-19, these are the examples which are released from these sewage pipelines or the joint wet markets, generally where the animals are slaughtered or animals are sold. So this slaughterhouse treatment is it is enforced by the many agencies in order to reduce the environmental contaminations. And there are the regulations given by the Environmental Protection Agency, then the slaughterhouse by-waste thermal treatment given by the Canadian Food Inspection Agency, and then pathogen disease transmission and ethical regulation by the European Union. But in developing countries and in most of the places in our country also, these solids or the liquid waste, they are directly disposed into the water bodies, and it creates the pollution problems. This is because of the inadequate water management and the treatment facilities. But in case of the developed countries, there are the modern habitats are exist, which are typically enclosed buildings where the well established waste management plants are available, where the blood is collected for the fertilizer production as well as for the blood meal. Then the solid waste are separated for the animal feed or the disposal. And there are the carcass incinerators also there, which can directly incinerate the carcasses and produce the ashes that can be used as a fertilizer. So this large amount of water which are used for the cleaning purposes, which was mixed with the oil, fat, and then these are immiscible, and because of that it get floated. That's why they have the mechanical pressure treatment or the pre-treatment system that separate this slaughterhouse waste, and they have also the fully automated system to recycle this animal waste. So basic objective of the effluent treatment is to convert this effluent into readily reusable resources, and as well as to produce the product which is safely discharged into the wastewater or the sewage system, and conservation of the water and nutrient, conservation of the soil, plants, animals, as well as the these water bodies, so that it can eliminate the threat of the disease. So generally there are three types of treatments given. That is the primary treatment, where the physical removal of the solids. Then the secondary treatment, it mainly include the biological treatment or sometimes the biochemical reactions. And the final treatment, where the after treatments, the disinfections are carried out, and finally it is released into the water. So we will start with the liquid waste management. In this, the water which are used for the rinsing purpose, they are continuously collected, and that can the slaughterhouse should have the well-built drainage system, so that this waste can be collected in some places. It should not be mixed with the or directly disposed of into the municipal drainage system without carried out the anaerobic or the aerobic treatment of the slaughterhouse waste, and then only we can dispose of or the throw into the this drainage system. But the uncontrolled, uncollected blood, they make a nuisance, particularly because of the clot formation and choking, and the septic tanks. Because of these are readily decomposes, and they are the ideal medium for the bacterial growth. And that's why it is very important to collect these bloods rapidly, so that the maximum potential of this blood can be utilized. If the slaughterhouse is having the concrete floor, so it will be beneficial to collect the blood and cleaning operations. So how we will give the pre-treatment or before going for the anaerobic digestion? So pre-treatment is nothing but the separation of the solids in order to enhance the availability of the substrate for the microbial digestion. And that's why the reaction time and the kinetics will be increased, and the total biogas production will be increased. Generally, the compactors are used for this purpose, so that it can use for the transport, then the de-water, compact all the remaining solids, and in order to minimize the moisture content. And these solids can be go for the anaerobic digestion. So this pre-treatment includes the catch basin, homogenization, equalization, then the flotation and settler, and it can reduce up to 30% BOD and 60% solids. Second application is the land application, where the directly biodegradable material can be throw away, and it can have the very good nutritional value. But the temperature and geography of the land affects the these operations, because in winter or in the temperate area, because of the winter season, these physicochemical or the chemical reactions takes place, or the biological reactions takes place in the slaughterhouse waste. They are minimum, and because of this, many problems are arising, like the odor, then the aesthetic problem, sometimes contamination of the groundwater, pathogen release. These type of problems are arising. Second treatment is the physicochemical treatment. This is the very prominent treatment for the solid separation, and after that, this can go for the secondary treatment. So primary treatments includes the dissolve air flotation method, then the coagulation method, then the electrocoagulation method, flocculation and sedimentation. Some of these methods we will see. And before that, the American Public Health Association, they made some standard methods to measure the pollution parameters. So in this, the chemical oxygen demand, biological oxygen demand, so total suspended solids, then the volatile suspended solids, nitrogen, fat, oil, grease, and color and turbidity, they can be helped to have the pollution parameter. So what is the biological oxygen demand? It is the demanded by the microorganisms for the oxygen. So whatever the sewage content, the biodegradable material, or the we can say the organic material, for the degradation of this material, the amount of oxygen which is utilized by the microorganism, these are measured, and this is on this basis we can have the idea for the digestibility of this slaughterhouse waste. So it can be obtained by the measuring the amount of oxygen consumed by the aerobic organism when a known volume of the effluent is added to a known volume of oxygen saturated water and incubated at 20 degrees centigrade for 5 days. And this is generally determine the concentration of pollutant remaining after the treatment, that is before discharging to the water bodies. So average BOD value for the poultry meat plant is around 1100 to 12.
Sir, you are not audible, sir. Dubal, sir, you are not audible. Dubal sir, you are not audible, sir.
Sir, voice is not coming. Please check.
Yes, we have been. I am informing the speaker. Dubal sir, can you hear me? Audience, just wait. Maybe some internet problem is there. Just wait for 2-3 minutes. Hello. Just wait for 2-3 minutes. Dubal sir is joining again. Just wait for 2-3 minutes. Dubal sir, you can start again, sir. No problem. You can share.
Hello, am I audible now?
Now you are audible, sir. Now you are audible.
Some internet issue came, that's why, sorry.
No problem, sir. No problem, sir. Sir, please share again, sir.
So, is it visible now?
Yes, sir. Yes, sir. Visible, sir.
Yeah. So, sorry for the disturbance, some network issue came here. So, beside this, some pollution parameter like the suspended solids, temperature, turbidity, and color volatile solids also helps in the to understand the which kind of treatment we have to give it. So, we will start with the primary treatment. In this, actually the solid particles, these are removed by the metal screener. Here, you can see this is the metal screener where the solids particles are trapped, and then after that the heavier particles they are settled in the grid chamber, and then it will go for the primary sedimentation. So this is the primary sedimentation tank where the effluent is coming from this left side, and this is suspended, and then the solid materials are we can say it is collected at the bottom side, and then the clean slaughterhouse waste it is passed further for the secondary treatment. So in this another treatment, that is the dissolve air flotation technique. In this method, some kinds of the micro bubbles, they are inserted into the effluent plant so that it can mix with the or it can give the agitation type of process, and so that hello.
All are requested to keep mute your mic. Don't talk in between. Sir, please continue,
Oh,
sir.
yeah, it will help to mix the small particles in the sewage water, and then the fat and then the grease and oils, they are coming upwards so that it can be float and it can be skimmed so that it can be removed separately, and the suspended solids, they percolated down and that can be separated later on. So some kinds of coagulants are also added, like the ferrous sulfate, ferrous chloride, aluminum sulfates. They are added to increase the efficiency of the removal. But ferric chloride and the aluminum sulfates, they are commonly used. In this method, the COD removal can be efficiently achieved up to the 30 to 90%. Then the BOD removal up to the 70 to 80%. So what happened? These coagulants are the positively charged, and they are bind with the negatively charged particle, particularly the amino acids and the organic materials which are generated in the sewage water. They are the negatively charged, and then they are trapped with this coagulant, and because of this, the sulfates and the chlorohydrates are formed, and that get precipitated and they get settled down, and so that it is easily we can remove it. Another method, that is the electrocoagulation, nothing but it is the same, where the electric current is applied. This is also one of the effective method. Another method, that is the membrane technology. We all know, everybody have at their home, we have the filtration, water filtration unit. So this is the same kind of where the reverse osmosis, nanofiltration, or the ultra filtration or the micro filtration techniques are used, and which can use to separate the solids or the minute particles, and then the sewage water will be clean. So secondary treatment, this is nothing but the biological treatment. Here the organisms are utilized for the digestion of the solid waste or the remaining after the primary treatment. So these microbes utilize the organic matter, and they are either do it aerobically or the anaerobically. But the currently biological and the thermochemical processes, they have gained tremendous attention from the industries, because these processes could recycle large amount of bio waste in a short period of time. So this biological treatment is nothing but the conversion of bio waste by living organisms, and thus it reduces the BOD concentration in the sewage water, and it removes the soluble organic compounds. So it removes the organic as well as eventually the pathogenic organisms, and reduction of BOD up to the 90% can be appear in this biological treatment. So this treatment includes the anaerobic or the aerobic or the facultative lagoons, as well as the trickling filters. And but the anaerobic digestion and the composting, these are among the very well-known technique for the slaughterhouse recycling. So anaerobic digestion, actually this is carried out in the absence of the oxygen. That's why it is called as the anaerobic digestion. And obviously, as it is carried out anaerobically, that's why the enclosed system is required. But here the fast reduction or the fast digestion of the organic material takes place, and with the production of the biogas, and it requires the BOD higher than the 2000 milligram per liter, then it will become advantageous. And pH the 7 to 7.2 is very important to maintain, so that faster degradation of the this sewage waste, that is the organic material, takes place. Here the complex organic materials are digested to the simpler compounds like the volatile fatty acid, and then it further digested and produce the methane and the carbon dioxide. In well-developed and modernized slaughterhouses, these high-loading anaerobic reactors, these are used. This is of two types, that is the batch reactor and the continuous reactor. The batch reactor, the name itself, the batch, that means here the batch wise the slaughterhouse waste are carried for the reaction, while in the continuous reactor a small amount of slaughterhouse waste are added for the recycling, or we can say the digestion of the this slaughterhouse waste. So this digestion is carried out in two ways, that is the mesophilic and thermophilic. Obviously, mesophilic involve the bacteria which can sustain the temperature between the 20 to 45 degrees centigrade, and this run up to the 15 to 30 days. But in case of the thermophilic, the bacteria can sustain the temperature up to 49 to 70 degrees centigrade, and it runs lesser period of time, that is up to 14 days. Obviously, thermophilic, as the name suggests, it is required, it tolerates the high temperature. That's why it produces a large amount of methane production and faster throughput and better inactivation of the pathogens. These are some of the advantages of this thermophilic digestion. One study stated that the bovine enterovirus and parvoviruses, this can be killed completely in the thermophilic digestion, but they can survive the mesophilic digestion system. The anaerobic digestion technology is being utilized the bio-waste, and simultaneously it produces the methane, as well as it has several advantages like the reducing the carbon dioxide emission, then the odor and the reduction of the pathogenic diseases. It also saves the land space. It produces around 48 to 89% the biogas, which results in the methane production. Then the 47.87% CO2 and 2.43% the ammonia. So this is the anaerobic digester, where the influent is coming from here, and then it is digested, and then the gas are generated, which are collected here in the system. So this anaerobic digestion, they have the high COD removal, low sludge generation. The sludge which is formed in this anaerobic digestion, they have the very good agriculture value, and it also helps to produce the biogas. So in some cases, the pasteurization step is also involved before the anaerobic digestion, so that it inactivates the pathogens, and it helps in the digestion system. But the digestion occurs in the 4 stages, that is the hydrolysis, acidogenesis, acetogenesis, then the methanogenesis. So this anaerobic digestion, they are carried out in the different way, like the anaerobic baffle reactor. So here the baffle are used, and this inflow of wastewater is coming from this way, and because of this baffle there is a reaction time is increased, and so that this is the sediment occurs in the first, second and third compartment, and then from this because of the increased contact time with the organisms, we can say the solids are helped to settle, and the air is flow further, and then the clean this water or the sewage is coming out, so that the up to COD removal up to the 90% of this aerobic baffle reactor can be carried out. And this is the similar anaerobic filters can be used here in this system, where the this is the somewhat similar to the baffle system, where the contact time is increased, and so that the whatever the reaction takes place, which helps in the settlement of this sediment at the bottom side, and clean water will be coming out after the anaerobic digestion. Beside this, some lagoons, we all know this system is we can see in the rural area. These are also help in the digestion. This is generally carried out in the areas where the winter or the cold season is there, and here the method is same. The solid material settle down, and this helps in the anaerobic digestion here, while the floating material, they are brought to the surface of the water, and where the anaerobic digestion takes place and the algal formation takes place. These are also help in the digestion of this one. But the disadvantage of these lagoons, that because of the light penetration minimize, the aquatic flora can be disturbed in this system. Another system is the anaerobic sludge blanket reactor. This is the single basin where the all operation, that is the feeding, reaction, settling and decanting stages are take place in the same reaction. There is some modification, that is the upflow method, upflow reactor, where the influent is coming from the bottom and that get mixed and there is a sludge granules form, and then the gas is collected in the gas chamber that is at the top of this system. So this is also another method. Besides this, the composting, this is the very common which we can observe, or we know it is familiar in the rural settings, and this is for the waste like the manures, litter, feathers that can be used for the composting. A large scale poultry waste can be mixed with the different ratio of the bulking agents like the cardboard, agriculture waste. They can help to mix with these manures and then helps in the digestion. So ultimately it helps in the soil fertility, and it is rich source of the carbon dioxide, hydrogen, this water and NH, that is the nitrogen compounds. But here the temperature, whatever it is reached during the composting, it can kill number of pathogens, but some organism cannot be killed during decomposition, particularly the endospores of the Clostridium bacillus, and that's why the anthrax rearing or the anthrax prevalent area, they should avoid this composting. So pH exceeding 8, that means in composting the pH is also reaches up to the 8, which also helps in the inactivation of the pathogen. Another method is the thermochemical processes, where the pyrolysis, liquefaction, incineration and gasification, these methods are there. Obviously the name itself, that is the thermo, means the heat is applied to sanitize the slaughterhouse bio waste, and then the chemical processes are utilized in order to degrade the organic molecules. This is highly recommended by the Canadian Food Inspection Agency. This is the technology where the intermediate liquid products are produced and which are ultimately helps in the production of the hydrocarbon biofuels and the petrochemicals. It is conducted at a high temperature in low oxygen concentration, while the gasification, this is the method where the partial air is utilized. But the problem of these techniques, it produces the greenhouses, like the greenhouse gases like the carbon monoxide and the nitrogen. So this is the some pyrolysis method. Besides this, the incineration method, where it is a well-known method where completely these carcasses are digested completely, and it produces the ash as well as the CO2 and oxygen. Gasification is also a method where the temperature reaches up to the 800 to 900, and it completely produces the decomposes the organic matter. It produces the carbon monoxide, methane, carbon dioxide, such type of gases. The poultry ash, particularly, it is produced during the gasification. It can remove the cadmium-like heavy metals, that is the toxic metals. Rendering is another method where generally this is a useful method for collection of the this melted fat or tallow, or it is also used for the carcass meal. This is conducting using the mechanical processes, including grinding, mixing, pressing, decanting and separating, and then the thermal processes for the cooking and evaporation, and the finally the drying. So chemical processes like solvent extraction can be done in this rendering plant. So coming to the aerobic treatment, that is another method of the secondary treatment where the name itself aerobic, that means the aerobic organisms are utilized for the disposal of the waste, where the organic carbon get break down into the carbon dioxide and the nitrogen, and this is generally carried out before the discharge or discharge into the water bodies. That means after performing the anaerobic digestion of the slaughterhouse waste, we can go for the aerobic treatment. It removes most of the residual BOD and it oxidizes ammonia and hydrogen sulfide to a less harmful nitrate and sulfate. So two types of methods are involved in this method, like the activated sludge process and trickling process. So this is the aerobic treatment where the bacteria, which are aerobic bacteria, which are used for the elimination of the organic compound, and depending on the amount of the slaughterhouse waste, or we can say the potency of the slaughterhouse waste, the time and temperature combination will be adjusted, and uh, it includes the variety of setup like the activated sludge, rotating biological contractor, anaerobic wastewater treatment, like that. So many aerobic treatment. So like so many treatments can be carried out. So this activated sludge process, this is nothing but the same like aerobic treatment process. It involves exposing of the effluent to the air and free floating microorganism, which includes the bacteria and protozoa, which digested these remaining material which are present after the anaerobic digestion. This method is very cost effective, and it removes the soluble and insoluble organic matter after the anaerobic digestion, and generally the absorption and the oxidation, these are the two mechanical mechanism of actions took place. So this is the activated sludge process. And another method, that is the aerobic sequencing batch reactor. This is same, but here the sequencing, the reaction is carried out in the sequencing manner. Like first it is filled with the sewage water, then it allows to react by mixing, then the solid materials get settled down, and then it is decanted, and whatever the remnant, that is the ideal, that can be separated. So beside these, the constructed wetland, this is also we can observe in the many rural setting. This is an attractive alternate, and where the biological and physical as well as the chemical processes that occur in these wetlands, and obviously the photosynthesis and the absorption and photo oxidation, then the filtration, this type of reactions takes place, and it helps in the helps to produce the fertilizers for the these plants, or the, we can say, the production which are we are taking from the agriculture operations. Another method is the alkaline hydrolysis, which represents a relatively new technology for the disposal of animal waste. In this method, actually, sodium or potassium hydroxide, they are utilized to catalyze the hydrolysis process, that degradation or the catalysis of the protein material or the carbohydrate material, what is the remnant, into the small peptide or the amino acids or the sugars or the salts. So in order to accelerate this hydrolysis process, it is usually conducted with the pressure and temperature. So this is the pressure and temperature that can be utilized. This is the trickling filter method. This is the generally deep bed, and it consists of the porous media, like this porous media, and above this porous media, this sewage water is we can say sprayed by the trickle, and then that gets percolated through these rocks, which consist of the organisms which are attached to the this media, and then clean water is coming out at the bottom, and it can be, we can throw it out directly into the water bodies. This is the sludge digestion. This is also the similar, where the solids are separated, then it will go for the further processing, and then the secondary digester take place, and finally it goes away. Then after pre-treatment, primary treatment, by the removal of the solids, semi-solids, then it will go for the secondary treatment, where anaerobic digestion and aerobic digestion takes place, and after that, whatever the remaining, that is called as the last remnant that can be disposed of directly into the land, or we can directly dispose into the water resources. So before disposing, we have to give some kind of treatment or disinfect this by utilizing the chlorine, potassium permanganate, or food grade acids also we can use it. And this can directly throw into the sea, river, land treatment, or in the oxidation pond or ditches or the aerated lagoons. So this is the oxidated ditch here. This is the aerated lagoons where we can directly add it. So this is the flow diagram for the effluent treatment plant. This can be used in the modern slaughterhouses. Actually, this is the photograph. I took it from the Al Kabir's Export Private Limited, where they have designed this type of treatment where we can separate the solids in the screen chamber, equalization chamber, then it will go for the primary digestion, then the secondary digestion, the gases which we can collect it, then it will go for the aeration tank, and then clarified secondary extract that can be go for the sludge or manure formation. So this way, these are the treatment which are carried out for the slaughterhouse waste management, where we are separating the solids as well as the digesting the organic, inorganic materials, and ultimately we are producing the slaughterhouse waste which is of free from the pathogenic organisms and which are safe for the disposal directly into the water bodies or drainage system. But before concluding, I would like to say we have to utilize these slaughterhouse resources or the animal resources. As we see, there are so many advantages of this waste, like we can produce the high quality effluent, biogases, fertilizers, these nutrients arising from this byproduct that can be used for the pet foods, animal food. But in the recent years, because of the problems of the BSE, that is bovine spongiform encephalitis, this feeding of meat or bone meal or the pet foods which are produced from these large animals, these are prohibited in most of the European countries. This fat obtained from this low cost material which can be used for the grease, animal feed, soap, candle, biodiesel, tallows, which can be used as a raw material in the steel rolling industry. We can go for the biogas plant also. There are so many advantages of the blood which can use for the fertilizers or the pharmaceuticals as well as the biodiesel. In Europe, animal blood has a long been used to make the blood sausages, blood pudding, biscuits, bread. In Asia, we are using it is for the blood curd, blood cake, blood pudding. It is also used as a emulsifier, stabilizer, even color additives. Besides this, many blood components such as fibrinogen, fibrinolysin, serotonin, immunoglobulins, these are also we can recover, which has the pharmaceutical actions, and blood plasma also can be used for the cook ham, hog hot dog, as well as the gelatin which are released from the bones. They have the medicinal values, pharmaceutical values. Even sausages can be prepared. Tallow and lard also can be used for the deep frying. Bones can be used for the neat food oil. So we have so many advantages of these animal resources. So we will utilize these, and then whatever the remaining after all these byproducts, that can be treated for waste, anaerobic or aerobic digestion, and then finally we will throw into the water body. Of these produce the slaughterhouse waste, which is the safe for us, safe for the environment, safe for our children, safe for our plant, safe for our animals, as well as safe for everyone. With this, thank you very much. Thanks for listening all. Thanks one and all.
Thank you, sir, for such a nice and exhaustive presentation. So now I will request the uh, participants, if they have any questions, so they can ask their queries.
हो गया.
Thank you very much, Dr. Zunjar Dubal sir, for your enlightening speech and excellent presentations. You have covered all the points regarding the effluent treatment plant for disposal of solid and liquid slaughterhouse waste. Although I know you are busy in that neighbor's funeral program, but still you have given your valuable time for the this very informative presentation. So thank you very much, sir. Now there will
Thank
be,
you. Thank you.
yes, there will be one or two questions from the participants. So I request
Yeah.
participants, you can ask directly or direct Dr. Vivek. If you have questions, please you can read.
Hello.
Sir, one question is there.
Hello.
Sir, good morning.
You can
Good
ask.
morning. Good
Yeah,
morning. Yes, tell me.
it's Om Prakash Patil. Sir, I have one question that you
Yes.
have just shown us the diagram, the flow chart of effluent treatment plant. I saw that one chamber look like a gas collector. So is it
Yeah.
possible to produce a biogas from ETP sludge or the odor comes from the ETP water? That gas can be collected in a biogas chamber, and we can utilize for to generate the energy for for generator or somewhere else. Is it possible?
Yeah, of course. I shown one slide, particularly in the anaerobic chambers. Uh, there are the chambers which are make specially for this purpose only, and where the after anaerobic digestion, some gases are released. They are captured by the some kinds of the. They have the instrument by that they are captured and they are utilized. So like the nitrogen, even these greenhouse gases, they are separated. Even some gases have been collected in this during this operation. So it is possible you can collect and you can translate it into the some other purposes also. After all, it's a energy you can utilize, no problem.
Okay, and sir, I'm sorry, I'm just asking a second question. Just allow
No problem.
me because I need some information. It is really interesting and it's knowledgeable for us. After the treating a water, in a slaughterhouse, we need X amount of land to release that treated water. So, is there specification that X amount of land you require to release that treated water?
Yeah, it's a very good question. See, every slaughterhouse, there are the norms when we are designing the slaughterhouse. Depending, that's why I have shown on my second slide. It is depending on the livestock unit. How much slaughtering you are doing in a year. So based on the everyday slaughterhouse waste, how much amount of plant or we can say the land is required for that effluent treatment. It is measured based on your production or slaughtering capacity, and then you can design accordingly. So it is entirely depending on your slaughtering capacity of the plant.
Okay, okay. Thank you so much, sir. Thank
Any
you
other
so much.
question?
Thank you.
हेलो
Yeah.
हाँ डॉक्टर दुब्बल सर, सबसे पहले तो मैं आपको बधाई देता हूँ कि आपने इतना अच्छा प्रेजेंटेशन दिया एफ्लुएंट
Thank you.
ट्रीटमेंट
Thank you.
के अंतर्गत। जैसा कि आपने बताया कि हमारे हिंदुस्तान में करीब 1175 या उससे ज़्यादा भी लीगल और इलीगल स्लॉटर हाउसेज कंडक्ट हो रहे हैं, तो 75 आपने बताया कि मॉडर्न स्लॉटर हाउस हैं। तो दो प्रेजेंटेशन है जो आप सजेस्ट कर रहे हैं वो तो मॉडर्न हाउस के लिए, स्लॉटर हाउस के लिए तो ठीक है, लेकिन मैक्सिमम संख्या इलीगल स्लॉटरिंग की है, जिसमें बूचर्स अपने घर पर ही 1 यूनिट से कम या उससे ज़्यादा स्लॉटर करते हैं। तो मैं ये चाहता हूँ कि आप क्या सजेस्ट करेंगे कि वो उनके द्वारा जो एफ्लुएंट निकलता है उसको लोकल लेवल पर ट्रीट करने के लिए, क्योंकि इसको अपन बंद तो कर नहीं सकते, क्योंकि एक बहुत बड़ी मात्रा है इलीगल स्लॉटर हाउस की, है ना। उसमें आप क्या सजेस्ट करेंगे ताकि हम कम से कम अपनी सॉइल और प्लांट्स और व्हाट एवर जो भी है वेस्ट मटेरियल की वजह से जो पॉल्यूशन होता है उसको बचा सके, एक जनरल।
Yeah, this is again very good and important question. See, in a small sector or we can say जहाँ पे एक दो स्लॉटर होते हैं दिन में, वहां पे एक्चुअली एफ्लुएंट प्लांट डालना फिजिबल ही नहीं है. बट यस, इन दैट केस जो कुछ रेमनेंट बचता है, जो कुछ छोटे छोटे पीसेस बचते हैं, कुछ भी, उसका हम कंपोस्टिंग करके, सिंपल कंपोस्टिंग करके, दैट कैन बी यूज एज अ फर्टिलाइजर. और वो हम एक वी कैन से जो डॉग मिल्क बना सकते हैं, डॉग को खिला सकते हैं, कोई हॉट फर्मेंटेशन, हॉट ये करके, थोड़ा सा हीटिंग करके, जो हम जानवरों को खिला सकते हैं, पोल्ट्री को खिला सकते हैं. जो वाटर बचेगा उसको अगर सिंपली हम क्लोरिनेशन भी डाल देंगे या पोटेशियम go for the land filling instead of draining into the river, you can go for the land filling. I can say that will be better to avoid the contamination of the river or the water bodies. That can be done, that is possible.
Hello, sir. I have a question. Hello.
Yeah.
Yeah, this is
Yes,
Dr.
yes.
Shankar Lingevada here.
Yes.
Yeah. How to remove odor
Yeah,
or
Dr.
dilute the odor which is generated around the ETP plant? How can you
How to remove?
remove it? Are there any technology developed for this?
No. It is very difficult. See, you cannot remove the odor in the slaughterhouse itself, but the drainage, whatever the waste is coming, that if your drainage system is enclosed, that can directly go to the TPP plant. So there it is possible. You can collect the odor. My first Dr. Om Prakash asked me the same question. So there you can collect these gases and that can be used as a biogas or the different gases. But in the slaughterhouse, it is not possible. In the drainage system, you can make the enclosed drainage system go further, and then the plant, the effluent plant, which is completely enclosed. Like in the one slide I have shown, in the particularly in the developed countries, they have their own collecting there in the effluent plant. So that is only possible.
Oh, no, my particular reference is, when the equalization tank, can we do something to rather remove that odor near that equalization tank?
Dr. Gowdaji,
No. Yes.
I will add in Dr. Dubal's answer.
Okay.
Saying that you can see here in India, so whatever the integrated slaughterhouses are there, mainly which are mainly meant for the export purpose. Okay, export oriented slaughterhouses. You can just walk through from the gate itself. Okay, you cannot make out there is a inside slaughterhouses. Okay, such technologies are available in the all integrated slaughterhouses in our country. Those are producing the meat, hygienic meat for the export purpose, even also for the domestic purposes. But only thing is that what Dr. Dubal is wanted to tell you that, see, whatever the waste which are produced in the slaughterhouses, okay, either it is liquid waste, solid waste, if they are properly utilized, economically utilized, okay, they will give number one the economic returns to the plant, so that whatever the overhead expenses are there, they will take care of all the waste which are produced in the slaughterhouses in the form of the rendering plant or the ATP plant, everything it will take care of. Okay. Only
In
thing
other
is
words,
that you
reducing
have
the
to
load on
use
the
it properly, economically. And in if your ETP plant is very good, there's no problem of any smell and nothing. Okay, all the sections of the ETP plant is work properly for the degradation of all the organic matters which are present in the slaughterhouse waste. so that it can be converted into the value-added things, like you will get a clear water. That water you can use it for the gardening purpose, washing the vehicles which are coming by taking the animals from the outside. Okay, so for that purpose, you can that water you can also have the aquaculture also in your slaughterhouse. You can culture the fishes, different fishes. So that water you can use. And what are the solid material manure we can use? It is high quality manure which can be used for the gardening purpose, agriculture purpose. So there is no problem of any smell or nothing will be there. Okay. So only thing is that we have to plan in a such way that we can utilize each and everything. So that is why it is a say in slaughterhouses, except the last cry of the animal, everything it is utilized. Okay, in the slaughterhouses. That is why that the people they make profit once they have set up a good quality slaughterhouses in our country. Okay.
Yes, sir. Yes, sir. Thank you.
I think now, thank you very much, sir, for a very excellent presentation. Now
Thank you.
we have another eminent speaker, respected Dr. S.B. Barbude, sir. And sir is working as a director, ICR National Research Center on Meat at Hyderabad. And sir will present the topic on different hurdle technologies used for improvement of meat quality for export purpose. So now I request our professor Dr. R.J. Jhende sir to give brief introduction about our respected Dr. S.B. Barbude sir.
Okay.
Jhende sir, please.
Thank you, Dr. Vaidya. First of all, I thank Dr. Zunjar Dubal for giving a very nice, excellent speech about what are the slaughterhouse waste and how they are managed in the slaughterhouses, what are the different technologies, everything he has covered. I can make out from the responses from the various participants for his lecture. So thank you, Dr. Dubal, for inviting and accepting our invitation for delivering today's talk. So now I have pleasure to introduce the another eminent speaker today, the respected Dr. Barbude sir. So Barbude sir is presently is a director, a dynamic director of ICR National Research Center on the Meat, Hyderabad. Dr. Barbude sir, he has completed his bachelor degree from the Nagpur Veterinary College, master in and PhD in IVRI in 1993 and 1996. And after that, he did his postdoctoral fellow at Justus Lieb University, Germany in 2006. Dr. Barbude sir, I think in the hall of 199 participant, I think everybody knows he is very dynamic and he is known by his work, hardworking and very alert. Whatever it is happening in the field of the division of the veterinary public health, I think that Barbude sir knows first. So that much I can say about his alertness and that very dynamic director. So Barbude sir has received many awards. To name some awards, he got an Young Scientist Awards of the National Academy of the Agricultural Science in Animal Science for the year 97 and 98. He is also the recipient of the Biotechnology Overseas Associateship Award in 2005-06 to undergo a training abroad under the Overseas Associateship Program of the Department of Biotechnology, Government of India at the Institute of Medical Microbiology in Germany. And he is also working as an expert member in the various national and the international forum. In the international forum, he is a member of the joint FAO-WHO expert committee on the microbial risk assessment, on a microbiological risk assessment of the Listeria monocytogenes. He is India's Listeria monocytogen man, I can say. And he is also the member of the scientific panel on the biological hazard of the FSSI, Government of India, New Delhi. And he is also the member of the International Advisory Committee of International Symposium. Under his able leadership, we have organized this ISOPOL at Goa during 2013. Then he is also the fellow of Indian Association of Veterinary Public Health Specialists. Research projects he has handled there are about 32 institute-funded projects and extramural 9 projects sponsored by the various agencies like ICR, TBT, DST. And with this, we can think about his research career. Okay. Then these are scientific research contributions since 2006 in various areas of the or aspects of the veterinary public health. And he has published about research papers in the international journals 117 and book chapters contributed 19, and he has published and authored books. There are 5 books. Okay, so you can see the depth of his research in the different aspects of the veterinary public health, of the food safety, or you can say the microbiological molecular microbiology. So with this short introduction of Barbude sir, I invite him to just deliver the talk on the different hurdle technologies used for the improvement of meat quality for the export purpose. So I invite Dr. Barbude sir, please.
Thank you, Dr. Jende sir. Am I audible, Dr. Jende?
Yes, sir. Yes, sir. Yes, sir.
Okay. So thank you for the introduction. And now, good afternoon, everybody. Just we are past 12 o'clock. So I will be talking on the hurdle technology, but meat processing, particularly for export purpose also, and also how this meat can be processed or preserved by use of the hurdle technology. So I come from the institute, which is a part of Indian Council of Agriculture Research, the ICR National Research Center on Meat, which is located in Hyderabad, and it is the only institute in India which caters exclusively to the meat sector. And we have various laboratories. Particularly, we have enabled accredited meat speciation laboratory. That is the only facility in India, we can say, and we get all our forensic samples and regulatory samples all over India. Also, we have meat processing facility, which is having APCC 22,000 certification, and so many other aspects. And we also work on residue analysis as well as microbial aspects of this meat. So, but today topic is something else, hurdle technology. So, yeah. So hurdle technology actually, you may be aware this is one of the technology or techniques, or you can get combination of techniques which can be used for meat preservation. So meat preservation, if you see, that is since time immemorial, actually see the environment originally also adapted by mankind to maintain the continuous supply of food throughout the year. So whenever there is excess supply of excess of meat, or if you want to store, maybe pickling, cheapest, you can see, or other aspects are drying of meat. So many aspects are there where actually this meat is being preserved for future use or used for using the difficult times also. Or if you want to go, particularly if you see your courses or everybody, they use the preserved meat like that. So also mankind began to preserve the food in times of plenty, that's I mentioned, and to tide over the scarcity of food. And so the primary purpose is the food preservation to prevent the food spoilage. Means the food actually. Now we are talking about meat, but you think of meat, milk, or any type of vegetables also. So the spoilage, that's so that if you store for longer period, it should be useful for it should be edible for any farm. So that is the main intention of this. And then the primary cause of food spoilage can be action of microorganisms such as bacteria, molds, or yeast, or any type of enzymes actually. Even if you keep in the sunlight, some enzymatic changes may happen. So primary cause of spoilage is just that prevent the action, unavoidable actions actually, or the action which is not required by us. Then microorganisms also can survive and develop only under particular microenvironments. So if you take care of those microenvironments, so you can preserve these meat. Then coming to meat preservation, preservation of red meat or poultry or their products is accomplished by the ensuring that their immediate microenvironment is favorable, unfavorable for the growth of microorganisms. For example, we bring any type of meat and keep in the refrigerator so that the organism's growth will be standard, and you will get that meat what you want actually. It will be can be kept for longer time. So also by controlling the action of autolytic enzymes, that's I mentioned. Then also preservation of meat can be also initiated by drying of meat either in the sun or on the fire, particularly smoking and also other type of particularly barbecue and other things. There are so many things that are there. So that can also methods of preservation for drying of meat or also other things. Then most of the processed meats available today originated from techniques that were developed to extend the length of between the slaughter of animal and the consumption of the meat derived from the animal. So we'll discuss what are the different techniques and so how it comes actually. So what are the different principles of food preservation, sir? Particularly delay in the microbial decomposition, or maybe delay in the self decomposition of the food, because the food also may have inherent microflora or some of the enzymes. Third one is actually the prevention of damage caused by insects, animals, mechanical causes, any type of, if you are keeping in open, that's why packaging systems they come actually. So to prevent damage from the environmental things actually. So for delay of microbial decomposition, particularly you are keeping handling this product particularly as far as the aseptic conditions. So not avoiding the contaminations, so that the surface or the slaughter process where you are used, then removal of microorganisms, particularly by filtration. So then handling by group by activity by low temperature, drying or deep freezing. Then also killing of the particularly heating, or for example in milk we use pasteurization. Also irradiation, which is also technique. The delay in decomposition, particularly blanching, we use mainly fruits, also vegetables, also other products also. Then prevention of delay by purely by chemical reaction, particularly oxidation means antioxidants and other things also are bacteriocides, or so many things you can add to them. So, so this is just summarizing the methods of meat preservation, particularly moisture control, temperature control, or direct microbial inhibition. So we have different techniques: drying, then freeze drying, or lyophilization, salting, curing, smoking, particularly moisture control. For temperature control, we have different low temperature, then chilling, freezing, or hot temperature, super chilling, or return processing, canning. Then for preservation by direct microbial, we see particularly irradiation, antibiotics, or chemicals, very organic acids, and so bacteriocides, so many things are there. So this is just to summarize all the methods. So what is the hurdle technology now? Comes that is hurdle. So as a name hurdles, hurdle, you can imply that somebody will also say, "I don't create any hurdles in my work," like that. So this hurdle technology is the same term actually. So it is a deliberate or intelligent use of the combination of existing or novel preservation techniques to establish a series of preservative factors, hurdles particularly, that any microorganisms present should not be able to work. So this is what this definition, this defines the hurdle technology. So it should be maybe combination of different. So what are these combination and what are different methods actually? We'll discuss in detail later. So it can be fluid preservative, it can be gas atmosphere, it can be temperature, it can be pH, it can be water activity, and also avoiding the harmful bacteria. So combination of hurdles area actually is what's it. So what is the need actually? Why we need hurdle technology? So why with the fast growing economy and more working women are now with the family system particularly, we want everything. So many women, they are working now, they want to cook food, and income is also increasing. So we take the food from supermarket or any online marketing or so. So it has increased, and the ongoing trend has to be eat out. So demand for processed foods particularly has increased in recent times, and their consumers also, they are demanding fresh, natural, and minimally processed, ready to eat food products. Also, so if you see the history actually, hurdle technology developed in 1980s and use the first shelf life stability of particularly sausages, and then they to improve the total quality of foods by application of intelligent mix of hurdles. So what are, we will give some examples also later, how this research in India and abroad also, they use these different type of hurdles to preserve this meat. Then hurdles can be deliberately combined to improve the microbial stability, sensory quality, food as well as the nutritional quality of. So with the use of hurdle technology, see, not only meat has been preserved, but this technology has also been used to prepare new and novelty products also. Low moisture products or shelf-stable products. So many type of products also they evolved by use of this hurdle technology in practice. So coming to the history, 1976 particular hurdle term coined by Listener and Rodel. And then 1985, the hurdle technology, all the principles were given by Listener in 1985. Then barrier technology was also exploited by hurdle technology by the same person. Also, multi-target preservation of foods was found by again Listener. Then invisible technology, was introduced by Listener in 1990 as a synonym to herder technology. So these are things that so one person actually this year, but throughout the world, including India, including I think Bombay Veterinary College also, they conducted a lot of studies for meat preservation, including this herder technology. So what are the principles actually? So first is actually disturb about the homeostasis of microorganisms, so that if you disturb the organism's homeostasis, the organism will not grow and this spoilage will be hampered. Then pathogens should not be allowed to cross the jump over the hurdles effect. So there are two criteria, as you say, process hygiene criteria and also food safety criteria. So both the criteria have to be allowed actually when you are producing in a ready to type of food or something. So both the criteria need to be followed. So you have to employ hurdles actually, which can take care of both the things. Then preservative factors should not allow the microorganisms to proliferate and so remain in inactive stage and even kill them. So this is also whatever the stages or whatever the factors you are employing, maybe lowering water temperature or freezing, whatever it may be. So it should not allow the organism to grow. Then also Hubner effect shows that the complexity of interactions of temperature, water, pH, humidity are important factors in microbial stability. So these have to be exact. So these are the important intrinsic or extrinsic factors for microbial growth that has to be taken care. So this is just through flow diagram. You ensure the safety of the food from microbes, prevent food spoilage, enhance the shelf life of food, reduce economic losses, and also control foodborne infection classification. So these are objectives, primary objectives. This I mentioned. This is one of the things also we can say safe preservation. Preservation may be there, but safe preservation so that after the preservation, if you want to use that for edible purpose, it should be edible. So that should be the thing. So what are the different principles? So you can use any type of physical hurdles or physicochemical hurdles or even for microbial, particularly if you see the physical, there are several examples. Aseptic packaging or electromagnetic energy, so many microwave and so many things are there. Modified atmosphere or packaging films or photodynamic inactivation or ultrasonication. So there are several ways where these actually physical hurdles can be created. Apart from that, if you consider physicochemical, then carbon dioxide or ethanol or lactic acid, lactopurify systems, low pH systems, then use of sodium nitrate, or sodium potassium sulfides, or spices and herbs, or even surface treatment agents. Spray particularly we use in many type of carcass washing also. So these are, you can say, all consider particularly physicochemical hurdles. As far as the microbial decontamination is concerned, you have different type of antibiotics. Then we have different bacteriocins. We have competitive microbial, lactic acid bacteria, other things. Then also protective cultures also. So bacteriophages and so many things, they are coming for this purpose. So you have to consider one or all activities. So you have to formulate, or you have to see what type of product you want to storage, how long you want to storage. So depending on that, you can just summarize that what type of hurdles can be created. So different parameters, you can use high temperature, you can use low temperature, you can use reduced bacterial activity, you can use increased acidity, high acidity, high acid foods we call it them. Then you reduce the redox potential, then biopreservatives and other preservatives. So if you see the nutshell actually, these are the techniques actually we use in routine purposes. Even any type of food which is of animal origin, high temperature, particularly pasteurization, if you see, low temperature, freezing, refrigeration. And if you want to particularly, how long you want to store, that's why others are told, how long you want to store the meat, particularly. If you want to store for 1 or 2 days or 3 days, just refrigeration is enough. But if you want to for export purpose, what we will be discussing later also. So you need to how long it can, because it will be shipped from one country to another country. So it may take 1 month or 2 months or 3 months. So maybe up to 1 year actually. So you have to use a deep freezer, freezing or -20 or -18 degree temperature, so that it can keep longer. Then reduce activity, particularly because water activity is very, very important for microbial growth. So you are reducing the particular dried foods. So if you are reducing the water activity of the food actually, so that the bacteria becomes inactivated and cannot grow. So that can be also used. Then increase acidity, particularly pickling and other things comes out of that. Then including redox potential in the particular bacterial growth is very important. Biopreservatives, I told about bacteriocins and other things also. So this is actually hurdle used in food preservation, particularly any type of food, including meat. So these are the cycle already told. These are the things actually: temperature and water activity, pH, electric potential, and other preservatives you can use. And these hurdles can keep spoilage or pathogenic organisms under control, because microorganisms cannot overcome all of these hurdles. So if you keep only pH, so something may happen. There are only reduced activity if you are not packing nicely and keeping in exposure to air. So some contamination may come. So you have to use the combination of hurdles. That's why the combination is important actually, so that any type of heating or low water activity or lowering pH or canning or using different aspects also. So you can use different type of hurdles to such products. So this is just I already narrated about this. So it can improve the food stuff quality and ensure the microbial activity. Then food remains safe and stable for longer duration of time, and high sensory and nutritive value of the can be also can be preserved. Applicable to both small and large scale industries. It can be used for anybody, any small or bigger scale, and does not change the composition or integrity of food. That is more important. So this you have to when you are selecting any type of hurdles, you have to see that the composition and integrity of the food is not altered. Then also it fulfills the current demands of the consumers for fresh, natural, and minimally processed foodstuffs, and it is also economically suitable for the nation, as it saves money, time, and energy, and other resources also. So now, what are the different aspects? So I mentioned earlier also one of the biggest problem actually is the homeostasis. So disturbing the homeostasis of the microorganisms by various hurdles, it results in the death of the microorganisms. And it's one technique you can use for food preservation by achieving disturbing the homeostasis. Then this is the best way to do this: deliberately disturb the several homeostatic mechanisms simultaneously. So you can use stress conditions or low moisture or multi-target preservation or sourcing. So by this way, the particularly this homeostasis of the bacteria will be affected. Secondly, actually metabolic exhaustion. For example, we keep the modified atmosphere. So if oxygen is required for bacteria, so that will be exhausted. You are putting nitrogen or carbon dioxide. So it will what will do? It will you can say auto sterilization of food products. Also could be achieved by metabolic exhaustion. So whatever required for the growth of the microorganisms also. So our spores. So you have to do metabolics here, exhaust the metabolism, so that the microorganisms in Hunter technologies they can have repair mechanisms also. But we have to take care of the different hurdles. So then there are different types of kinds of bacteria or molds or yeast which overcome and sustain the high temperature treatment also. So that is we call thermodynamic bacteria, or that's why the pasteurization temperature, particularly in milk, you have to decide what time high temperature short time or low temperature high time actually. And there are sterility are using in canning of foods also different things are. So that is all techniques you have to use at metabolic, so that the metabolism is completely stopped and the microorganisms will not grow. Third one is actually stress reaction. So some microbes require resistance and may become more virulent under stress conditions, and they can synthesize stress or we can say shock proteins, we call them actually. So there are several factors actually the bacteria can overcome. If you do the genome sequencing of bacteria, actually, you will find so many stress factors will be genes will be controlling only stress factors like pH or better even antibiotic resistance. So many things they come actually. So these different responses of the microbes under stress conditions might hamper the food preservation. So that's why we have to see also what type of stress if you employ for in technology, so that the bacteria can be completely inhibited and it will not able to grow. So exposure to multiple stresses simultaneously activates the energy utilizing the synthesis of several stress proteins, in turn making the microbes metabolically weak. So this is how these when you are selecting different type of products, you have to see for this purpose. Then multi-target preservation of food. This is also very important. So hurdles applied in the targeted food material might just have effects on the microbial stability, but they could not act synergistic. So that's why again this is also principle. That's why we have to use the hurdle different type of maybe lowering pH or lowering activity also, or a different enzyme the degradation. So many things simultaneously. So when you plan for any type of hurdle technology, you have to take care of all those things. Then coming to this is again just Common hurdles. This I already mentioned, pasteurization, water activities, salting, curing, which is one of the hurdles. Blanching, I already mentioned, freezing, modified as per packaging, then pH, preservative, refrigeration. So already I mentioned about this thing. So now just some technologies I will just pasteurization, particularly milk industry. They are very much rely on the particularly this technique and refers to treatment of to destroy the microorganisms of public health significance. So all pathogens cannot be destroyed, but finite organisms they will be completely destroyed by this technique. And also using the industry to kill the microorganisms in the food, and also pertinent pathogens also reduce the spoilage organisms. So in milk industry, there are very much familiar with this term, STAT and LTAT, LTLT. So these are around 74 degrees Celsius for 15 seconds, high temperature, short time. So this is very common technique used in the milk industry. Then refrigeration. It is common household also. We are using this technique ideally 0 to 4 or maybe up to 8 degrees Celsius. And for short-term preservation, maybe up to days, we cannot say weeks also, and it slows down the microbial growth, or respiration, or enzymes, or chemical reactions. And so it can lower the rate of chemical reactions, and also action of enzymes. And usual temperature cold storage are 4.5 to 7 degrees Celsius. So this is everybody, even superstores, they use this technique, refrigeration particularly. Then freezing. So for freezing, is the ideal temperature is around minus 18 to minus 20 degrees Celsius. And it is used to any type of export houses particularly. They use these techniques, blast freezers, and also they if you are storing a longer time also, they have to use this temperature only. So there should not be then thawing or evaporated freezing and thawing. So the quality of products depends on the time and also temperature. So if you are storing for minus 80 degrees, and this is now what the export industry follows now actually. So and then freezing is a long-term preservation, and it can slow down the chemical reactions, also stop microbial growth, also respiration. And when stored at minus 30, the enzymatic and microbial changes may be stopped or extremely slowed. So the many times the question I actually ask, when you deep freeze at minus 18, will all the microorganisms should be killed? So no. So that's why actually even if you want to eat this actually frozen meat, you have to cook it thoroughly. Otherwise, some of the bacteria, when you keep it refrigerated or bring it room temperature and keep it, some bacteria may again get regain actually. So this is very important actually. Then high pressure processing. So it is used for post processing technology and application of high pressure up to 400 MPL increases the microbial protein solubility and improves the water retention of food products. So this is also can be used to produce the healthier meat products. So particularly it can have pathogen elimination, then shelf life extension, and then clean label products, reduced sodium products are convenience. So many things and advantages are there using the high pressure processing. Coming to the packaging, everybody knows actually this is most important technique actually, where if you go any type of supermarkets, you will find any type of product, they are packaged, very attractive packaging. So it is also one of the gimmick for actually marketing also. So it is a crucial for maintenance of quality, protection against damage, and also microbial contamination. So we can have vacuum packaging, you can have modified atmosphere packaging, you can active packaging, or even having intelligent packaging. So particularly now in mid sector now, this smart packaging or intelligent packaging, more and use actually this is being coming actually. So the consumer, by seeing the only packets only, he can know whether the meat is good condition or spoiled. They are keeping some indicators, some strips they are keeping. So by the seeing the change of the color itself, they can know that whether the meat is of good quality or not. So these are different techniques actually which can be used for packaging things. I will not go into detail because the one technique takes a lot of time of discussion. So these are just I want to mention that these are different type of techniques for use for packaging. Then natural antimicrobials. So there are so many organic acids. There are so many protective cultures, particularly bacteriocins, lactic acid bacteria. Then antimicrobial microbial origin, particular lysozyme and cytosine. Antimicrobial peptides also. We are also working on antimicrobial peptides, particularly whether it can be used for food preservation. Then a lot of phytochemicals, plant antimicrobials. we can, their spices and herbs are there, which are actually brown spices also are herbs, are smoke from wood. So all these things are there. Again, the bacteriophages. Now, a lot of research actually going on actually on use of bacteriophages. Even some of the bacteriophages, they have been licensed by FDA also as a food preservative, particularly Listeria already licensed by FDA in US. So many things are there. So if you want particularly fermented products or other type of products, so this type of hurdles can be used and different type of products also can be prepared from this. Then irradiation. So this is also good technique actually. And even Bhabha Atomic Research Center, I think Bombay Veterinary College also they worked with BARC in Mumbai to use this irradiation, particularly for preservation of meat. So ionization addition of usual doses of 1 to 3 kilogram or up to 5 kilogram. So they are using, and even though if I'm talking of meat, but even when you are exporting mangoes and other things, this irradiation actually they have units in Bangalore and other areas also, when they actually irradiate so that the fruit fly and other problems can be solved. So this is a very versatile techniques. So this can be used also for type of meat preservation. Then intermediate moisture foods. This I was telling earlier also. So this using of hurdle, many type of new type of meat products also they are being produced. Particularly intermediate, we call it intermediate meat products. And the binding of water that's present preserve the intermediate moisture. So this reduces the availability of water for the deteriorative reaction. Also, water can be mobilized by adding permissible humectant additives such as glycerol, glycols, and sorbitol, sugar, and salt. And you can impart such type of peculiar taste also type of the meat product. So they possess bacterial activity is very less, 0.6 to 0.85, and this enables the food to be able to store at room temperature because the growth of microorganisms can be inhibited. So this is the technique actually where different type of hurdles you can have specialty products also using this. Then coming to the effect of humectants, also roasting, lithomy chemicals are several properties in jerky made spent in meat. So these are also some of the products. These are made using the different type of hurdles in the meat industry. Then biltong, it is one of the South African product actually, intermediate moisture food, which is can be prescribed as intermediate moisture food. And moisture is controlled. Then different type of other hurdles. Two types of major importance produce microbiological stable and safe biltong, namely salt and moisture content. So using these two hurdles actually, this product has been made and also battery activity has been reduced. So that this is a new product has come using the different type of hurdles. Then what are the applications in the meat and meat products? So I also mentioned earlier, so there are several studies carried out in the globe and also in India. Treatment particularly see here combination of refrigeration, heating, and acids, curing, then also combination of newly applied technologies. Now newer just I mentioned about the smart packaging also. Then natural antimicrobials in combination with irradiation. This is one fellow has used earlier in 2003 itself. Then combination of irradiation and pH reduction, also organic acids. They are also one of the researchers they are used. I think Dr. Bride is from Bombay Veterinary College only. He used treatment of sheep and goat using propylenic meat, lactic acid, acetic acid, and also bacillus cereus, that flora, to inhibit this flora. And reductions were greater than the eradicated meat than without acids. So this is this type of study also carried out in India also. Then marination of chicken legs in natural plant products. So this is also be used for the different type of product, including irradiation have been used. Then another promising combination hurdle, particularly extending the shelf life of fresh meat. High hydrostatic pressure combined with bacteriocins. Then bacteriocins suspensions pressurized with high pressure technology. Then use of mixing is very common actually. These antibacterial by different active bacteria, bacteriocins that produced by those things. So this is also being used actually. High pressure HSP, then lysine, injured cells, that they have been also used under lactic acid bacteria. Then also HSP treatment with mechanical record poultry meat. Particularly, they are used the pressure of around 200 ppm and lysine, bacteriocins also. So these are actually some of the examples where the people have used the different type of hurdles for preservation of meat. Then also water activity, lowering of the water activity and pH as variable hurdles. Then shelf-stable curry also, which is different type of product developed with the combination of salt, pH, other preservatives and packaging material. Then also charky mix to observe the possibility of development of enterotoxinic, RES and clostridium. So they also use different type of hurdles. Then irradiation, reduced water activity and vacuum packaging, particularly to prevent the growth of clostridium sporogenous. So these are actually some of the examples that I wanted to give how these different type of hurdles they were used to preserve different type of products and also prepare the newer type of those products also. Then how this now our topic actually is export promotion. How this technology can be combined. So high moisture foods, processed foods with water activity more than 9, we call them actually high moisture foods. So they can be chilled or frozen. So maintaining low temperature is energy consuming costly. Therefore, the temperature above the storage can also reduce the stability and so. So therefore, besides low temperature, additional hurdles such as heating or lowering pH or redox potential or water activity or preservatives or competitive flora, this can be applied. For example, just I mentioned about bacteria, these bacteriophages. So our country still we are yet to come with such robust technology for this purpose. But whatever the meat is exported, particularly from India, buffalo meat, are worth actually around almost 25,000 crores every year. So mainly they use deep freezing, -18 to -20 degrees Celsius. So this is the most important why they use technique. And then they pack actually nicely. Packaging will be there, cardboard or other type of packing. So external contamination can be avoided. So then another type actually fermented meats, also important process actually, particularly using the different type of lactic acid bacteria. A lot of research, yeast also. Lot of research is going on, particularly on making the fermented products because these are being prepared by different type of people and different countries actually. So this is being also evolved. But as far as export from India is concerned, hardly we export any processed meat. Hardly maybe 1 or 2% maybe poultry products only made. But hardly we export any type of products which make up buffalo meat. Maximum chunk comes from the actually buffalo meat, frozen buffalo meat itself. So in particularly common farming purpose is also sausage. So they are making now pork sausages also, other type of sausage also, but still the not much export from India. And these ST products will also be developed to improve the keeping quality without chilled and frozen meat. So this is possible, but as I mentioned, the energy consumption, other things also, and then if you want to transport actually, that time also a lot of expenditure has to be involved. Then deep chilling and super chilling are partial freezing. So this is also actually one of the techniques. Our institute also is working on this aspect, super chilling particularly. So freezing the meat at a sub-zero temperature, just 1 degree below the where the meat freezes. So this can be uniform ice crystal can be found on the surface of meat, which insulates the product from minor temperature changes during storage. So almost we have also developed one super chilling cabinet also at our institute, and this one. So lot of energy can be saved with this. So maintain the freshness of the meat for a longer time, and up to a week or maybe up to 15 days. If you want to shorter period, I think this technique can be very handy instead of going for deep freezing. Then also another type is smoked and cured meat products. So the fermented I already mentioned. Then this type of smoked and cured meat products also, but still we are not able to much export much type of this type of product. Fermented techniques are available, technology is available, but still we are not making dent in this aspect also. So though the processing meat industry in India is I think almost growing around 20% every year, but still the overall meat processing, if you see, it comes around hardly around 10% and maximum from poultry, around 7% it comes from the poultry sector. But that's why we need to have particularly small ruminants, also buffalo meat products actually. That is the area of research also for the industry. Then I was mentioning about the packaging also earlier. So different type of packagings now they are coming. So they are nanotechnology based things. They are being used actually. Chemical sensors and then biosensors they are being used. Then also now barcoding labels are RFID tags So lot of countries they are using, but our country as still we are in the just in the we can say stage of infancy itself. And another thing which is lacks actually in the export purpose, particularly the traceability. Traceability now we are also working also government of India taking. So such system actually particularly it has to be certain with now APIDA, which export actually the regulatory agency for export purpose. They have from mid.net, you must have seen actually. So post slaughter traceability is there. But with this traceability, you can say what from where the meat has come, how it has come, then how it was, when it was processed, and how it was labeled. Everything can be single barcode can be created. So such technology now we need to work in India itself also. So these are actually these slides will actually what different type of systems we can use, like moisture regulators or ethylene removal systems or carbon dioxide scavengers or antimicrobial active packaging things. So many type of packaging have come now actually. So this is also one of the things you can say type of hurdle actually, different type of packaging we can use. And properties you can have, you can interact with food or release or absorb substances, particularly volatiles from food. Also extension of the shelf life. Also doesn't act on food. It should monitor the conditions of the I mentioned about. We are also working on smart packaging. So we have also developed some nano sensor. Just you have to keep on inside the package. And if your meat is spoiled actually, the indicator the color changes. You can know what type of when the meat is spoiled or is in that good condition. So these things can be easily monitored. Then this is only what exactly what we are doing with smart food packaging. So just inside the trap, you have to keep one indicator. So indicator color changes means you can know what type of whether the meat is of good quality or bad quality. So the packaging can help in monitoring the quality of products during shipment. Also minimize food wastage. Also prevents foodborne infection. So this is technology is coming on. A lot of people they are working. Even the industry of packaging, we want to work with them also on this aspect, particularly smart packaging for foods of animal origin. Then shelf-stable foods, as I mentioned, particularly pickles, things are there, where the hurdle technology is the basic concept of shelf-stable foods. So with us also our institute, so many entrepreneurs, they have signed agreement to work on particularly pickle. They bloom the chicken pickle. These are one of our entrepreneurs. Also some of the other people also they are working on this aspect, and they're ready to eat foods products. They do not require refrigeration also, so energy saving will be there. So shelf-stable products which can be stored at room temperature. So products like pickle, meat cookies do not require refrigeration. So even now different type of cookies from the millets and other things are coming. Like that you can deal with the meat also, so that the shelf-stable product can be come. It will be the hurdle is particularly low moisture content. Other type of bacteria also can be used for such technology. So use this hurdle technology. Different variety of the shelf-stable products can be produced for marketing. So to just to conclude my talk actually. So today's public health concern is particularly the minimal damage to product with maximum protection to food to microorganisms. So any preservation technique lead to damage to nutritional value or sensitivity. So that's why hurdle technology is the best way by which we can improve the both of these characteristics. Also, it can be ambitious goal for optimal food preservation, and then including the food of animal origin, and it can encompass the target multi-target preservation of food utilizing various factors as hurdles. That's I mentioned actually. So if you want to any type of products or what type of application you want to have, or what type of product you want to take to market, or which type of product you want to export, or what type of marketing product you want to market, so you have to decide the hurdle. There are different. So hurdle which is not actually you can say it's not a rocket science, but combination of all this is just I mentioned also. So there are different type of preservation techniques. So just a combination of things are there. So that one hurdle may not use, for example, refrigeration. But if you use a bacteria refrigeration, you can store the meat for 1 or 2 days more. So like that, just you have to see that a different type of preservation techniques can be combined and used as a preservation technique. Then also it can be used in an appropriate way with a good tool for achieving taste safe and tasty food product of high quality. I mentioned earlier also. So you can come up with a newer novelty product also with this. If you want to have different type of products, smoke products, and pure products, and self-stable products, you can use different type of products. Then minimal sensory and nutrition changes it affects actually using the other technology, and which makes the product more valuable and acceptable than obtained by the conventional methods. Also, this is valid for modern processes preservation, also for the traditional foods. Even the traditional foods, you can also have like Hyderabadi Halim is there already. They are sending during the month of Ramadan, or Hyderabadi biryani also they are sending now to other countries also using this because it cannot be stored longer. But different type of hurdles they use and they send it. Then this also make its way longer shelf life of foods of animal origin to fight against the malnutrition and hunger, even the deficiency. So whenever actually there excess is there, any time of the prices of the meat falls in the market, particularly poultry. So they can use different type of products and refrigeration or freezing, and they can keep this meat and they can sell when the prices are up good that available in the market. Then also it can be intelligent mix of the hurdles which makes work synergistically, which already I mentioned. So I thank also my colleague Dr. Yogesh Ladekar, who has helped me to prepare this presentation. So this is what I wanted to share with all of you. Thank you. Linda Saab, over to you.
Thank you. Thank you. Sir. Thank you so much. In mainly preservation of meat and meat product for the domestic as well as for the export purpose. You are highly elaborated the topic to the understanding to the all the participants. I also thank showing your the activities of the laboratories also. So how this NRC meat is also helping the meat industry to solve their what are the rectify their problems of the meat industry they are facing for the export of meat. Sir, we have now a question answer session, and I ask Dr. Vivek Shukla to take this question answer session, which are mainly written in the chat box also and directly from the participants. Vivek.
Thank you, sir. So I request the participants to ask their queries. They can ask directly or they can write in the chat box so that we can ask to the sir. So any question?
I think there is no any question in the chat box. Only thank you for your best, excellent presentation. These are the comments you have received from the various participants across the country. So now looking at what are the contents of the presentation, really it was very nice contents and very informative to the participants. So directly any participant is interested to
Sir, one of the participant is asking why high hydrostatic pressure is useful with the meat products. So what is its significance like that he wants to ask?
Yeah, see, actually, this is one of the techniques where actually these homeostates are also metabolic weight actually, particularly they can be disturbed by the that spoilage actually. Even this solubility I mentioned about also solubility in the different proteins, other things that also can be achieved with the high hydrostatic pressure. So the different type of things are available, and how to pressure and how much pressure to be used. That depends actually what type of product you want to just store, how long you want to store.
Thank you, sir.
Thank you,
Any
sir.
other question?
Hello.
हाँ बोलिए बोला पाटिल साहब
I request participant please introduce yourself. You tell your name and your concerned company name or your designation, and then you ask questions.
Hello.
हाँ बोला.
हाँ सर, बोला बोला.
Dr. Bhagutai, good morning. And this is Dr. Sunil Kulkarni.
हाँ बोला था. नमस्कार साहेब.
Namaskar. Just one question on the bacteriophages. Is this bacteriophages, they are commercially, they are working as a for the decontamination of the carcasses, and is there any adverse effect on the human health or anything, any that type of studies are made internationally or in the India?
Yes, I think this is already, as mentioned, the FDA has already approved the particular listeria for spraying purpose, and they found there is no adverse effect on the human health. So that's why carcass contamination, decontamination already. So India actually many people are working on this aspect, but India will still have to actually license this use of bacteriophages, particularly in the meat preservation. We also want to work. Now we'll be tying with a institute in Hyderabad who was already working on the bacteriophages. I hope to come out. But in US, yes, it is already licensed by US FDA for meat purpose.
Thank you, sir.
Thank you.
One more question. One more question if anybody want to ask.
Dr. Jende, if you are no question, I will give some 2-3 minutes about our institute. Dr. Jende?
Yes, sir, yes, sir, yes. Please, please.
Yeah, I think those who are aware of the NRC meet is okay, but those who are not aware actually. So we are having agribusiness incubator at our institute, and those who want to become the entrepreneurs in the sector, meat sector, right from the farm to fork, livestock farming to the processing or marketing. We are the people who help these prospective entrepreneurs, and also we conduct a lot of training programs for these prospective entrepreneurs. Also, we have one of the best laboratory for meat processing with us, which is the FSSC 22000 certified. And anybody, the incubator, who wants to have their own product, they have, or if they want to have the product from us also, we can have hand holding. If you don't have facility, you can come to us, use become our incubator. You can use our facility for maybe period of 1 year. You can test market your product. You can develop your marketing. You can develop your marketing strategy, and just you can expand your business. So that is the offer I can give you the people who are online. So take this opportunity. Secondly, we also have the nodal agencies, particularly from under post-tech from FSSI, particularly food industry test these trainings. We are also one of the nodal institute identified by MSME in the meat sector. So if you have any very good idea with you, come to us. We can help you to get hand holding with this MSME or other type of organization. Even the managed incubator is there in Hyderabad. With them also we can give tie up. Our NAR board is under ICAR sector. So you can come to us. We'll to getting the
Doctor is Babu, Director of Telangana.
Hello.
वो चलते रहता क्या?
हाँ जी, कुछ क्वेश्चन पूछना है?
हाँ सर. Yeah.
नहीं कोई.
So thank you so much. Really, it is. I can also endorse what the respected Barbudya sir has said about his institute. Really, it is a very nice institute to visit and see what are the different technologies the institute of industry adaptation. And I am also requesting sir, and I already requested before also sir. Sir, we have to give some slot for sending our student there, and also some of the faculties from our university for undertaking the hands-on training program in the different aspects of the food safety, so that we can train them as our university and our institute has, and also the MOU for undertaking some of the collaborative work and collaborative activities to be done between your esteemed.
Yeah, Dr. Yes, have already if some faculty want to do winter school also with us, from 8th of February. So they can join. We will extend the separate program for the faculty of MAPSU or any veterinary subjects.
I hope. I think some internet. Now it's okay, sir.
So definitely will help for this, and whenever the programs are there, even if you have a group of entrepreneurs, let us know, but not before March. It should be after March only, because already we have planned the training programs for February and March. So from April onward, definitely have the group of entrepreneurs, group of students, definitely will or even faculty will arrange.
Yes, sir. Yes, sir. Okay. Thank you. Thank you so much, sir. Thank you.
Yeah. Thank you. Thank you very much, sir, for your very informative and motivational and inspirational speech, and your valuable time you have given in spite of your busy schedule. So thanks a lot, sir.
Thank you,
Now I
Dr.
declare that today's session is over. So tomorrow, I request all the participants, please join at 10:50 a.m.
Hello.
Yeah, day after tomorrow. Yeah.
Sir, I'm requesting for the 1 minute. I just want to ask something to sir. Can you please allow me for a minute?
Please go ahead.
Yeah, sir, Om Prakash Patil here from Pune. I just want to ask: is there any training program for non-veterinarian, you can say, participants or candidates who is interested to know more about the diseases in animals or poultry sector? Because what happened in the small sector slaughterhouses, that promoters or owners can't be offered to recruit a full-time veterinarian, but yes, they can recruit someone like the technical person. For that, they want to send them for training to do a like AM or to identify diseases, basic disease in the poultry or animals. So is there any training program can be planned for them?
Yeah, Patil sir, see, we
Yes,
have
sir.
programs, but see, identifying of diseases and other things is a specialized job of veterinarians.
Oh, sir.
Entrepreneurship and other things, we have the people from IT, maximum people, they come around 60% people will only from engineers only. So for processing sector and other things are basic information, because we deal with the actual meat sector. So for this, there is no, it should be not be veterinary, any 12th or who knows actually little about this, can do business. So they can come for training. But as far as disease is concerned, actually, so I think this is a specialized job. By seeing the 1 week or 2 weeks, nobody will learn that all the diseases, this livestock or poultry they encounter actually. So they have to take some help of the veterinarians only, or some of the specialists as a consultant or so, whatever it may be. But meat processing and entrepreneurship, yes, we can entertain anybody, need not be veterinarian for that purpose. Okay.
Okay. Dhanyawad, sir.
Yeah.
Thank you, Jhanti sir.
So
Thank you.
if there are no questions, I declare that today's session is over. Thank you, sir, and thank you all the participants. Thank you so much.
Thank you.
Thank you very much, sir.
Thank you, sir. Thank you.